Hello, everybody. Today we have the pleasure to have with us Dr. Marie Passet from the Laboratory of Hematology of Hôpital Saint-Louis in Paris, France. Marie, it's a pleasure to have you with us. How are you today?
Hi, well, I'm great and I'm happy to be there and discuss my work today.
Thank you very much for accepting our invitation. And we are going to talk today about your research article recently published in Blood entitled Concurrent CDX2 CIS3 Regulation and UBTF-ATX and 7L3 fusion define a novel high-risk subtype of B-cell ALL. I'm telling you, like right now, I'm not gonna repeat anytime soon this fusion gene name. It's really a trouble for me. So first of all, can you give us a little overview of the genomics of adult B-ALL, that is the topic of your research?
Yes, so before focusing on adult ALL, it's important to know that B-ALL are classified according to recurrent cytogenetic and molecular abnormalities such as high hyperdiploidy, low hypodiploidy, or chromosomal translocation leading to oncogenic gene fusion, for example, KMT2A rearrangement. And this alteration drives specific transcriptional programs and are associated with a distinct clinical presentation, with distinct response to treatment, and finally, a distinct prognosis. So cytogenetic and molecular characterization of B-ALL at diagnosis is really important, and it's useful for risk-adapted treatment stratification. That's really true for pediatric ALL because they are well characterized, but in adults, with the exception of Philadelphia chromosome positive ALL, which really benefit from a distinct treatment with the addition of Trainee kinase inhibitor, few genomic abnormalities are widely recognized as useful for treatment stratification. This is partly related to the poorer knowledge of genomic abnormalities defining a relevant subgroup of patients as compared with the B-ALL in children. And because the prevalence of ALL subtype varies with the age. For example, the most frequent subtype in children are high hyperdiploidy and autophasis wrong fusion, subtypes associated with good prognosis, but they are less frequent in adult. Autophasis wrong fusion accounts only for 2% in adult. So finally, until recently, about half of adult B-ALL cases was uncharacterized and so-called B-other ALL.
I see, thank you. And can you guide us through the design of your study? So what did you explore? Which kind of patient cohort in particular? How did you get these samples? And with which technique did you study these patients?
Yes, so to improve our understanding of adult B-ALL, we wanted to identify new oncogenetic subtypes within the uncharacterized cases, so within the so-called B-other ALL group. So we were looking for a new oncogenic alteration that drives specific transcriptional program. So to do so, we performed transcriptome profiling of a large cohort of adult B-ALL, more than 300 cases harboring no cytogenetic abnormality using RNA sequencing. And we have also performed long-read whole genome sequencing for some cases. So with RNA sequencing, we can perform transcript fusion analysis to identify new fusion. We can perform unsupervised hierarchical clustering to identify cluster of patients harboring the same transcriptional program. And we can perform mutational variant calling. And with long-read whole genome sequencing, we are able to identify structural variants, for example, copy-neutral rearrangements that are missed by RACGH.
I see. So with all these techniques, you were able to find a cluster, right? That was like a new signal in all these variety and multitude of like so-called other B-cell ALL, right?
Yes. So first, with the RNA sequencing, we identify in our cohort subtype recently described, such as the DUX4 subtype or the subtype with the ZNF384 fusion, or a subtype defined by a point mutation of PAX5, the PAX5P ATR, and other group. And we discover a cluster of cases without recurrent known alteration. And so to elucidate the genomic alteration of this novel subtype of BALL, we perform long-read whole genome sequencing on only 2 cases of this cluster because it's quite expensive. And so integrated analysis of long-read whole genome sequencing and RNA-seq data showed in that specific cluster a deletion involving FLAT3 and PAN3 gene on chromosome 13. And this deletion is located upstream of a gene called CDX2. And this gene is one of the most highly expressed genes in the novel cluster. And briefly, CDX2 is the homeodomain transcription factor involved in development during early stages of embryogenesis, including primitive hematopoiesis, and it is not expressed in adult bone marrow. So here it's an ectopic expression of a gene involved in primitive hematopoiesis that have already been reported involved in acute leukemia. So we've got a very good oncogenic candidate to explain this novel cluster. But we got the surprise that the analysis showed a second alteration, a short deletion of 10 kilobases at chromosome 17, involving the UBTF and ataxin 7L3 genes. And the deletion leads to the loss of the end of UBTF on the intergenic sequence. And this resulted in an interim UBTF ataxin 7H3 fusion transcript detected in RNA-seq data and confirmed by PCR. And we next show that the association of these 2 alterations, so the deregulation of the CDX2 expression and the fusion of UBTF ataxin 7L3 was present in all the cases of this novel cluster, so we call it CDX2 UBTF ALL.
Oh, I see. So these 2 alterations in chromosome 13 and 17, pass me the joke, 13 and 17. So also like bad numbers for BALL2, right? But what is the genetic mechanism underlying the pathogenesis of this specific cluster of BALL? Can you guide us through the functional experiment that allowed you to outline how this aberration leads to oncogenesis and BALL?
So first, I'm going to begin with the easiest to explain. So the UBTF ataxin 7H3 fusion. So we have identified a deletion between these 2 genes. So the deletion generates a fusion transcript, and we show on Western blot that the resulting chimeric fusion protein was expressed in patient derived xenograft cells. So we've got an altered protein generated by this deletion and this fusion. So for the deregulation of CDX2 expression is the big part of the work. So we have identified a deletion upstream of the CDX2 gene using RSCGH, and we have found this deletion in all the patients. And so to better characterize this deletion, I have performed a capture-based sequencing on this specific genomic region from the CDX2 gene to PAN3. We identified the common minimal deleted region, which included the promoter and first exon of FLAT3 and BAD3. And next, I have analyzed the breakpoint sequencing and found recombination signal sequencing like motif. Motifs recognized by RAG enzyme active during lymphopoiesis to perform VDJ rearrangement. So the deletion occurred through off-target cleavage by the RAG recombinase in a differentiating B cell precursor. And such RAG-mediated deletions are frequent in ALL and occur in region of open chromatid. So I wanted to understand if those deletions were causative of CDX2 deregulated expression or just related to active transcription of the locus. So either the deletion leads to CDX2 high expression, either CDX2 high expression means that the chromatid is open at this locus and the chromatin is accessible for RAG enzyme. We analyze allele-specific expression of CDX2 using a single nucleotide polymorphism for understand if there is an alteration anywhere in the genome that leads to the expression of both CDX2 alleles, or if we have the expression of only one allele of CDX2 and a genomic alteration on this specific allele leading to CDX2 deregulation. And in every CDX2 UBTF case, with informative polymorphism, we have shown a clear monoallelic expression pattern. So next, we have performed sequential RNA-DNA FISH on a patient-derived xenograft of CDX2 UBTF ALL. So we have combined a probe to visualize the CDX2 nation transcript for the RNA FISH part, and two probes for the DNA FISH part, one mapping the deleted region and one as control mapping a region non-deleted. And we have showed that when we have the probe for the CDX2 transcript, we never had the probe mapping the deletion. So the CDX2 allele is always expressed from the allele that harbor the deletion. So the CDX2 deregulated expression was mediated by the genomic deletion upstream of CDX2. We next I've studied cis regulatory elements to understand what happened in this specific deleted region. For that, we've done chromatin immunoprecipitation and sequencing for histone modification in PDX of CDX2 UBTF ALL and in a cell line called NALM16. This cell line highly expressed CDX2. This cell line doesn't have the deletion at our patient, but have a duplication in the PAN3 gene. And so a CHIP experiment revealed an enhancer in the PAN3 gene, and this enhancer is never affected by the deletion in our patient and is located in the duplicated region of NALM16. Next, we perform 4C sequencing on NALM16 and other control cell line using the CDX2 promoter as the point of view. So 4C technology is for circular chromosome conformation capture that allow the detection of a genomic region that interact with a particular region of interest, here the CDX2 promoter. And the first step is the covalent fixation of chromatin in a close spatial proximity, followed by fragmentation, ligation, and digestion steps. And finally, DNA fragments are circularized and amplified by polymerase chain reaction with specific primer, here specific of the CDX2 promoter. And so 4C showed an interaction between the CDX2 promoter and the PAN3 enhancer region in NALM16 only, so in the only cell line that express CDX2. And finally, to better understand the role of this enhancer, we have designed a CRISPR interference experiment using single-guide RNA targeting the PAN3 enhancer, co-expressed with the transcriptional repressor domain fused to catalytically dead Cas9. So CRISPR interference is a technique that allows for sequence-specific reexpression of gene expression. So here we were targeting the PAN3 enhancer. So with our construction, we have put repressive chromatin marks instead of active ones. So we have turned down the enhancer in PAN3 and have looked at what happened on the CDX2 expression in NALM16. And we have observed a marked reduction of CDX2 expression at the transcript and protein level. So all these results demonstrate the role of the PAN3 enhancer on CDX2 aberrant expression. So to summarize, we identify a novel subtype of ALL characterized by the quite uncommon association of two genomic alterations. So the first one, the deletion between UBTF and ataxin 7S3 that leads to a fusion on the expression of chimeric protein. And the second one, a deletion involving PAN3 and FLAT3, is responsible for the hijacking of an enhancer in PAN3 that interact with the CDX2 promoter and leads to CDX2 high expression.
Wow, Marie, that's like incredible and so well detailed. Thank you so much, and congratulations really for like outlining so clearly the study design and the flow of your functional data. Another question, are these cases characterized besides, of course, like the culprit lesion, also by additional genomic or transcriptomic features, like specific co-occurring copy number alteration or so?
Yes, regarding transcriptomic feature, some OX gene were moderately expressed in CDX2 UBTF ALL, and CDX2 is an upstream regulator of OX gene, so it's quite expected. And gene set enrichment analysis showed enrichment in gene regulated by the polychondroexpressive complex 2 in embryonic stem cell and gene expressed in cord blood derived hematopoietic stem cell. And also negative enrichment in gene related to cell cycle and mTOR signaling, suggestive of lower metabolic state. And regarding the genomic landscape of CDX2 UBTF ALL, we characterize additional alteration using array-CGH and targeting sequencing. And the most frequent copy number alteration was the duplication of the 1Q arm seen in half of the cases. And other alteration involves the loss of a 6Q or a 9Q, or a deletion of PAX5 and its promoter. Targeting sequencing show frequent mutation of RAS pathway and CREB-BP genes. In addition, RNA-seq analysis showed 6 CR4 mutation, and that's a gain of function mutation. They have never been reported in acute leukemia, so that's what quite unexpected. And that's the same mutation as the mutation described in WHIM syndrome and Waldenström.
See, yeah, like in the congenital cases. So now let's go from the bench to bedside. What about clinical association? Did you notice any particular characteristics of this new cluster of patients you identified as compared to other BALL?
So we have identified 26 patients with CDX2 UBTF ALL, and 20 were women. So we have a highly skewed sex ratio. The immunophenotype of CDX2 UBTF ALL was frequently classified as pro-B stage. 17 patients were included in the GRAL trial, so it's a protocol prospectively enrolling adults aged from 18 to 59 years old. And so the female predominance was significant in this cohort. Patients tend to be younger, even if it's not significant. And blast infiltration in the bone marrow was significantly lower, and the immunophenotype pro-B.
I see. And any particular insight on the survival outcomes of this patient instead?
So this patient display a slow response to induction phase. So only half of patients have a peripheral blood response to steroid at day 8, and a low bone marrow blast clearance at day 15. 3 patients need a second induction to achieve a complete remission, and all but one evaluable patient had a high minimal residual disease. So CDX2 UBTF ALL is a subtype with a very poor response to chemotherapy. So with this result, patients receive more frequently allo stem cell transplants. Also, the cumulative incidence of relapse was already higher in this subtype in univariate and multivariate analysis. However, survival did not differ between CDX2 UBTF ALL and other BALL patients, which may, I don't know, reflect a favorable response to current salvage procedure in BALL.
I see. So it's from what I gather is important to identify this patient because they don't respond very well to the first line treatment. So it's important to know also and to identify them in order to quickly use appropriate treatment. Well, very amazing. All this study is like really shedding light on the shadowlands of adult BALL, especially the subtypes that are not as now so well characterized. So thank you very much for doing so. And can I ask you instead, why did you choose to study this particular aspect of hematology? Can you give us a little bit background on you? Why did you decide to study this aspect? Did you apply for any grant for doing so?
This study is, as you say, a part of a project that is really the whole characterization of adult BALL. The prognosis of young adults with BALL is still unsatisfying, and so it's important to improve our knowledge on adult BALL. So also with the recent evolution of technology has helped us with the gene expression profiling on microarray, or even more recently, whole transcriptome sequencing in the study using RNA-seq approach, allowed the identification of relevant leukemia subtype at the CDX2 UBTF that defined by their transcriptomic profile, and also enable the identification of gene fusion which represent the oncogenic driver. Now we on order perform RNA sequencing of BALL cases without recurrent alteration. Diagnosis, so it's quite a study from diagnosis and research. It's we work on both sides, and I have not speak yet about phyolite subtype, but some phyolite cases with ABL classes fusion really benefits also from the addition of tyrosine kinase inhibitor. So it's important to perform BLL characterization as soon as possible for diagnosis purpose and also for research purpose. So.
Thank you very much, Marie. It was a pleasure to have you with us, and thank you again on behalf of the EBMT trainee community and the committee to having showcase your research for us today in such a digestible format for all trainees. Thank you very much and enjoy the rest of your day. Bye-bye.
Thank you. Bye.